So thanks for coming along. It's much appreciated. So I'm going to talk about the molecular analysis facility, which is sequencing and bioinformatics, and I am John Cole. Okay, so what is sequencing and bioinformatics? Well, a typical bench project that could be a PhD or a whole postdoc, you might consider one gene or a small number of proteins, a small number of things. When you're doing sequencing, when you're doing omics, you can simultaneously consider for a sample the entire genome, so 3 billion bases, the entire epigenome. You could look at the entire transcriptome for each sample all at once, so something like 50,000 genes assayed in one go. You can do splicing, and you can now even do this at the single cell level with spatial information as well. So you could take 1 million cells and you could assay the expression of every gene in each of those cells effectively. And you could do that spatially, so you know which cell is sitting next to which cell and how that affects expression. And you can actually do this with 2D or 3D coordinates now. So it's come a long way. It's not even that expensive to do, and it's become a very routine type of thing to do. Of course, this produces vast amounts of biologically relevant data for you to kind of explore and, go off and publish and win your Nobel Prize for. You might produce hundreds of thousands and potentially millions or trillions of different observations of different numbers. And that requires computational skills and, of course, experience to be able to go in and analyze that and answer your research questions. I'll just skip through this, but sequencing is increasingly a ubiquitous part of the life sciences. So it's grown very rapidly. Geo, which is one of the big data repositories, has nearly 250,000 experiments on it and over 7 million samples. I think and probably most people would agree, it's increasingly unusual to find labs that have never performed sequencing or omics, and many labs do this habitually. It's kind of one of the main things they do. It's getting increasingly cheaper with a wider range of applications every year. So our facility, molecular analysis, has a long and esteemed history. So it actually begun all the way back in 1995, if anyone can remember all the way back then, with the Molecular Biology Support Unit, which was started by Graham Hamilton and Julie, who are still working with us now. And that's gone through several evolutions. The Henry Wellcome Functional Genomics Center, that got merged into Polyomics in 2012. Simultaneously, there was a bioinformatic facility set up in immunology, which I used to run, and that then got merged into the sequencing arm of what was Polyomics when Shared Research Facilities was created. So it's been around a long time. For any sequencing project that you would do, so it doesn't matter what you research or who you are or where you are, omic projects always have these kind of 5 steps of sequencing projects. So you, of course, have your sample preparation. You then have to do something called library preparation, where you essentially get your samples ready for sequencing. You do your sequencing, which is kind of like a big printer. What comes off that sequencing machine is effectively nonsense. It's millions or billions of sequences. They don't make sense on their own. There has to be some stages, some bioinformatics stages to turn that into useful, meaningful tables. So that would be your data processing. And then at the end, you've got your data analysis, where you answer your research questions and publish or get your thesis. So the question is, which of these stages is the most important? And of course, you need to do all of them. I can understand the argument, which a lot of people do say sometimes, that if you don't prep your samples, you can't do any of these steps. But the inverse is true. If you prep your samples and you do everything up to here and don't bother to do the analysis or don't have the skill set to do that, then you really haven't done an omic experiment. You have nothing useful at that point, nothing usable. You've just got expensive data gathering dust on a hard drive. So essentially, plan to get to the very end. What we can do, how we can help you with this, is we can advise you on the sample preparation, what works, what doesn't work, this kind of thing. How many replicates might you need? We can do a power calculation. And we can help you, we can do all of these stages. So we do the library prep, we do the sequencing, we can do the data processing, we can do the analysis. And we can help you with analysis all the way up to the point of figures and text for your publication. It can be end to end. or we can help at any stage. Maybe you only want help with library preparation. Maybe you've sequenced elsewhere with collaborators and you only need data processing or only need analysis. It's up to you. We can help in any way you like. We've got an excellent team. So there are currently 6 of us and hopefully another one soon. So we're kind of split into 2 groups. So we've got Julie, Chilla, and Shelley, who are the lab team. So they do the library preparation and the sequencing. And then we've got David, Graham, and myself, who are the bioinformatic data analysis team. So we work together. I think David is probably, as far as I'm aware, the only person in the university who actually can do everything from start to finish. I suspect. We've got a huge amount of experience. So between us, we have 58 combined years of library preparation and sequencing experience. We have prepared manually over 12,000 samples, which is a lot. We have, that's a lot of experience. I think Julie has done over 8,000 samples on her own. We've supported over 1,600 projects, so individual research projects within the university and beyond. And we've got about 36 years of bioinformatic experience. So the real point here is that we have a vast amount of relevant experience. As a team, there's very little we haven't done. If you've got a project on an unusual organism or using a new protocol, we'll have done something similar at some point. It'll be fine. On that note, we were the first within University of Glasgow to do in-house microarrays, to do in-house RNA-seq, chip-seq, and the first to do single cell. So we've been leading this in Glasgow for a long time. We've got various bits of kit, so pictures of little boxes that do fantastic things. We've got a PacBio Vega arriving very soon, so that's a long read sequencer. That can do up to 25 kilobases. We've got the Illumina NextSeq 2000, which is our kind of workhorse high throughput machine that can do up to 2 billion sequences in one run. We've got 2 10x Chromiums, which is for single cell. And we've also now got the 10x CytAssist, which is for spatial omics. So that's single cell resolution spatial transcriptomics. We've got a couple of extra things like a cell counter, etc., which helps a lot with single cell. So I'm going to skip over this quite quickly, but for the library preparation step, what I'm really trying to point out here is that is a fiddly, tricky, and highly nuanced thing to do. Yes, if you're a wet lab person, you could buy the kits and you can do this yourself for sure. However, I'll point out that it is more tricky than it seems. And, we do this a lot and we know what can go wrong. We know where you have to optimize and balance things. There's all kinds of things like your initial QC, getting the correct fragmentation, which will be application and organism specific. When you prepare the libraries, the steps might appear simple, but mistakes can happen here. Things that you won't know until you get to the analysis and you've paid all this money and used all this time. Things like your cleanups and this kind of thing. And then, of course, you've got your library QC again, where you have to check the integrity of the libraries, of the samples. Even small errors in there can have big effects down the road. So what we're kind of saying is that with the experience that our team has, we can identify any issues and we can correct it pretty much immediately. Whatever quality, sample or amount or organism, we will have done something similar with success in the past. We're experts at handling samples, all samples, and particularly those that have a low quality or a low quantity of starting material, or even just challenging samples. I mean, the team if anyone can do it, the guys in the lab can do it here. Again, these experiments can be very expensive. Why risk it, particularly for single cell? Use an expert. Just use someone who's done this literally thousands of times, okay? For library preparation, experience really does matter. You wouldn't retile your own roof. If you had to retile your roof, you probably wouldn't do that yourself. You could learn, but you probably wouldn't. You probably don't go off and get a kind of apprenticeship in fixing your car. You wouldn't service your own car. Again, downstream analysis can take months. It's really dependent on the quality of the library. On top of this, we can also do things slightly cheaper because, again, we've got the kit, we've got the experience, and we get kind of good deals on reagents. So I'll just go through some of the applications. Feel free to email me or put it in questions in the chat. So firstly, bulk RNA-seq, which is the kind of workhorse. So bulk RNA-seq, that means we can, for any sample, we can quantify, get an expression value for every single gene in your organism. Doesn't matter what the organism is, we can do it. We can also do splicing, so we could get sort of up to 200,000 alternate transcripts, maybe more. We can do this for any organism. We can go as low as 5 cells. So if you give us 5 cells, we'll be able to do this. Or we can go as low as 10 picograms of RNA. There are kits for this that work well. And we can do all the kind of different flavors. So your poly A, we can do this on formalin fixed paraffin embedded tissue. We could do ribosomal depletion. We can also do things like, small RNAs or microRNAs, etc. We don't kind of just do the service and run away. We have a lot of support. We want to look after you and make sure you get what you want. So we do a lot of consultation and discussion. We are researchers, so we have a kind of collaborative research focused mindset. And we can do all the analysis like your differential expression correction for batch effects, clinical inference if needed, pathway analysis, upstream regulators, ibosort. We can do everything. Signatures, biomarkers, ROC curves. So for the analysis, we can really do whatever you need. And our prices for this are very, very reasonable now. So we have updated pricing. Thanks to the move into SRF. So we can do bulk RNA-seq for as low as £90 a sample. We can do that, and we have done that. More typically, we're in the kind of £120, £140 a sample range. And again, our analysis is very, very affordable. Okay, we're talking a few £100s to do a comprehensive analysis. We have epigenomics, which is a personal favorite coming from a kind of cancer epigenomics background. So we can do all your kind of ChIP-seq, cut and run for histones or transcription factors or methylation. So for example, we can take a protein and we can find where on the genome it is in a sample and how that has changed between conditions. So we could do that for any protein that there's an antibody for. We can do methylation, so is your genome methylated or not at CpG sites. ATAC, so how open or closed the chromatin is. We can do all this. And of course, we provide a similar quality of analysis for that. So if you want us to do the analysis, maybe you've got a multiome, so maybe you want to do RNA and something epigenome, we can cover that. We've got lots of experience in that. Of course, we do genomics and metagenomics. We get quite a lot of interest on this. So we can do things like de novo assembly. So if you have a new organism that no one has ever sequenced and you want to know how its genome is, we can sequence it, we can assemble it, and we can annotate it. Of course, we do resequencing. So more commonly, if you want to get mutations in a human or see how some bacterial strains differ from each other, we could do that, of course. So we can do the preparation, the sequencing, and the analysis. We can build phylogenetic trees, like in the bottom right here. We can do all that. In terms of metagenomics, we can help you with metagenomics. So we have a kind of small group of sort of PIs that do a lot of metagenomics with us. So that means we can look at a sample. It could be soil, it could be poo, it could be urine. And what we're trying to find out is what types of bacteria are in there and in what relative abundances, okay? So we can do kind of amplicon sequence variants. So it could be 16S, 18S, or ITS. And that can be viruses, bacteria, fungi, or plants. So the kind of idea is, here is my sample, what's in that? We can also do shotgun metagenomics, which is what is in my pot and how is its genome looking, kind of what does it taste like, which is a little disgusting, but there you go. So we can do that. And we have great success. So again, as I said, something that we can offer here is because we have so much experience and the team are kind of very, very careful, we are good with samples that are difficult. And that happens a lot in metagenomics. So if you have challenging metagenomic samples, we will do well with that, I promise. So nearly there. So just a couple more slides. Our next application is single cell. So we have single cell omics. We can do assay individual cells in great quantity. So we can use the 10x parse or Illumina kits. We can do this for RNA. We can do this for surface proteins. We can do this for ATAC, so for chromatin accessibility. And we can also do TCR and BCR. We can do this for fresh or frozen cells or nuclei. It can be anything from 100 to 1 million cells per sample. Again, any organism. And we've done this for a lot of kind of strange things. So we've done human and mice, we've done rat, ladybird, we've done cockroaches, dragonflies. We've done a lot of different things. And of course, we can support with the analysis. So we're looking a kind of price range for single cell. It's come down in price considerably recently. So for 2,000 cells, you're looking at about £800 per sample. If you're doing 5,000 cells, you're looking in the kind of £1,250 range, which is probably about half of what it would have been last year. And if you're looking for 10,000 cells, it's about just over 2,000 per sample. And again, our analysis is very affordable, and we can do everything you want in there. All your QC, integration, cell types, cell markers, differential expression, pseudobulk, trajectory, ligand receptor. We can do all that for you. We work with you back and forth to do that. Our lead time typically is from 2 weeks. We aim to be within 4 weeks, but it's often 3 weeks is kind of where we seem to average from when we get the samples. And our last protocol we have is spatial omics. So this is, of course, a kind of massively growing field. So what we have in-house is the 10x cytosys, the Visium HD, which in my opinion is currently the most powerful spatial omic platform on the market, in my genuine expert opinion. And that can do a subcellular resolution of less than 2 micrometers. You can do this on fresh, frozen, or formalin fixed. It's poly A, which means it's an entire transcriptome panel. And there's a slide coming out in the summer, which is organism agnostic. So you could do this on things that are not mouse or human, even multiple organisms at once, which is fantastic. So you could do skin and fungus at the same time. And again, we offer the analysis. We've got extremely good pipelines for analysis. We're also can support analysis for any type of spatial. So we do a huge amount of analysis for cosmics and geomics. We've got extremely good pipelines. I would go as far to say we probably have one of the most powerful geomics pipelines in the world. And that means it's cheap for you and you get a lot. Okay, so of course, there's just 2 more quick things. We can support you on your grant bids. So we can give you costs. So if you're applying for a grant, we can give you costs, of course. We can discuss discounts on generating pilot data. We can get vast, good discounts on reagents if it's for pilot data from Illumina or 10x. So talk to us about that for pilot data. We can advise you on design. We can give you figures and text towards your grant bid. We can analyze public data sets to help and support your grant bid. And of course, we have a power calculation service. So if you need a power calculation, we can do that. And of course, funding panels have commented very positively on our low costs. We've heard this back from panels and from our expertise and our training programs. So you can give your bid a bit of an extra edge. If you ask, we can help. So finally, I'll skip through this fairly quickly. We've had huge amounts of success with OMIC and R training. So we've had over 2,500 people on our training courses in the last 5 years. Our mean rating is 9.5 out of 10, which is extremely good. This is after hundreds of reviews. So what we have is it's courses that are 100% entry level. They are aimed to equip people who have never done omics with the skills and confidence and experience to do this themselves. It's even been mentioned in the House of Lords, of all things. Our courses break down into kind of 5 different kind of sub-courses, and we've got a kind of table on our website where you can kind of decide, oh, I'm wanting to do bulk RNA-seq. I need to do this course, and these 2 might be useful. So feel free to email about that. So on that note, I'll thank you very much for paying attention, and much appreciated. And any questions, please let me know.